Hello everyone. Today we're going to talk about regulation of gene expression. This is covered in the second section of chapter 28. So that is regulation of prokaryotic gene expression. So the concepts we want to talk about today in this section involve the Lac Operon. We want to talk about how we use both positive and negative control in the control of that operon, including regulation by cap. And we want to use the Trp operon as an example of how attenuation regulates transcription termination. So let's begin with reminding ourselves that you can regulate DNA to protein at any of these steps, but as we said in class, initiation of transcription makes the most sense. The cell is able to conserve its resources most effectively if we control the expression of a protein by initiation of transcription. We can, of course, regulate at any one of these steps, but regulation of initiation of transcription is a very common place for a cell to regulate the expression of its genes. So in bacteria, gene expression is regulated in response to changes in nutritional and physical environment. So for example, maybe certain sugars are present or absent, or certain amino acids are present or absent. So if sugar is present, the cell needs to be able to metabolize that. If certain amino acids are present, then the cell does not need to synthesize those. So the bacterial cells are very good at adapting to their particular conditions and turning on and turning off a whole series of genes. So remember, about half of the genes that bacteria express are clustered into operons, which are coordinately regulated. So we're going to go over some examples of that today. So typically the way regulation of transcription occurs in bacteria is that you have some kind of repressor that binds to a sequence of the DNA called the operator. So, and the operator is usually located near the promoter. So when we say this is the promoter in yellow, that's the -35 region there, and that's the -10 region there. And this big blue blob here is RNA polymerase. So remember, RNA polymerase has to be able to find the promoter in order to know where to initiate transcription. So the operator is usually somewhere nearby. So using the bacterial lac operon as an example, it's a good example because it combines both positive and negative control. So if a cell is given lactose, lactose is a disaccharide, and you need an enzyme to hydrolyze the glycosidic bond between the two monosaccharides of that disaccharide. And you need to be able to bring the lactose into the cell. So both the Lac Repressor Protein and CAP, which is the catabolite activator protein, it's a helix turn helix protein, those are both involved in regulating whether the Lac operon is on or not. So the Lac operon should not be on if lactose is not present. You don't need those proteins or those enzymes if lactose isn't present. But it's also shut off if lactose is present and glucose is present, because bacterial cells will prefer to use glucose. So if they are provided with glucose, they will use the glucose and they will ignore the lactose. So the signal has to be on if lactose is present and glucose is absent. Both of those conditions need to be met. So just as a reminder, here is lactose. It is a disaccharide. It's composed of galactose and glucose, and the glycosidic bond between them is a beta-1,4 glycosidic bond. The enzyme that catalyzes the hydrolysis of this bond is called beta-galactosidase. So that is necessary in order to free these two monosaccharides, the galactose and glucose, so that the cell can use them as an energy source. So the other thing that beta-galactosidase can do, though, is it can catalyze a transglycosylation where it makes this compound right here. This is a beta-1,6 allolactose. So this is an isomer of lactose. And it acts as an inducer of the operon. So, so beta-galactosidase can catalyze this transglycosylation. So when the concentration of lactose increases, then the concentration of this 1,6 allolactose will also increase. So the way that the Lac repressor works is that when lactose is added to the growth medium, the concentration of allolactose increases because the beta-galactosidase will catalyze that transglycosylation. Then allolactose will bind to the Lac receptor. So the allolactose is a ligand. It binds to the Lac repressor and causes a conformational change that removes the Lac repressor from the DNA. So once that repressor is removed, then the genes that transport and metabolize the lactose are transcribed. The repressor is removed, but you get the best response if CAP is also present. So the first gene to be transcribed is the Lacz gene, and the Lacz gene codes for beta-galactosidase, and that's the enzyme that hydrolyzes lactose to glucose and galactose. So this is a figure from our book. There is an animation based on this, so I'm going to go ahead and click on this link here to play this. I'm going to have to enable the Adobe Flash Player for us to see this.
E. coli can metabolize a wide variety of different nutrients. One such energy source is the disaccharide lactose. Lactose enters the cell through a transporter protein called galactoside permease. Once inside the cell, the lactose is hydrolyzed by the enzyme beta-galactosidase to yield glucose and galactose, which are then metabolized via common pathways to produce ATP. However, if lactose is absent, it's a waste of often scarce resources for the bacteria to synthesize large quantities of galactoside permease and beta-galactosidase. Consequently, they don't do so. Instead, they have evolved a mechanism which senses the presence of lactose and induces expression of these proteins only when they are needed. The genes for beta-galactosidase and galactoside permease are located next to each other in the DNA, along with the gene encoding thiogalactoside transacetylase, whose physiological role is unknown. This cluster of three genes is called the Lac Operon. Upstream of these genes is a site known as the promoter P, the DNA sequence where RNA polymerase binds and initiates transcription. In between the promoter and the Lac Operon is a DNA sequence called the operator O. The operator binds a repressor protein called the Lac Repressor. The Lac Repressor is encoded by the I gene, which is located just upstream of the promoter site. The binding of the Lac repressor to the operator prevents RNA polymerase from initiating transcription at the promoter, thereby arresting the expression of the Lac Operon. When lactose is present, beta-galactosidase occasionally catalyzes a transglycosylation reaction that converts lactose to 1,6-allolactose instead of hydrolyzing it to its component monosaccharides. This disaccharide specifically binds to the Lac repressor, and in so doing, induces a conformational change that causes it, the Lac repressor, to dissociate from the operator. With the Lac repressor gene gone, RNA polymerase can bind to the promoter and initiate transcription of the Z, Y, and A genes in a single strand of mRNA. The resulting transcripts are repeatedly translated by ribosomes, yielding multiple copies of beta-galactosidase, lactose permease, and thiogalactoside transacetylase. The first two proteins then mediate the import and hydrolysis of lactose. Occasionally, beta-galactosidase will synthesize a 1,6-allolactose molecule, thereby preventing the Lac repressor from binding to the operator. However, when the lactose has been used up, 1,6-allolactose is no longer produced, causing the Lac repressor to once again bind to the operator and prevent the expression of the Lac Operon.
All right, so let's close from here and go back to our PowerPoint. So here, again, this shows the gene for the repressor is located upstream. So the repressor is made and then binds to the operator. There are a few more details here that we want to add to this picture. So the inducer here is the allolactose. So the inducer acts as a ligand to change the conformation of the repressor so that it can't bind to this operator right here. And when the repressor can't bind to the operator, then the RNA polymerase is able to bind to the promoter. And then you get the transcription of the genes Z, Y, and A, which code for beta-galactosidase, permease, and transacetylase, respectively. So let's talk about CAP. The video doesn't say anything about CAP. So this is how we integrate the signal to be sensitive to the amount of glucose that's available. Because if lactose is present and glucose is present too, then we don't need to turn on the Lac Operon. So decreasing concentration of glucose in the cell causes an increase in cyclic AMP. So cyclic AMP acts as a ligand and binds to the CAP protein. And when the CAP protein has cyclic AMP bound, the CAP protein has a conformation that enables it to bind to a specific sequence on the DNA. So the way that this works then is, if we look at our DNA right here, the binding site for CAP is upstream, and again here's the promoter, the -35 and the -10, so that's where RNA polymerase is going to be looking for. Specifically, remember it's the sigma 70 subunit that's looking for that, and then there's the operator, and then the genes, the like Z, Y, and A will be here. So the operon is off if glucose and lactose are present. That's because there's CAP is not bound and the repressor is not bound. The operon is off, sort of doubly off, if glucose is present and lactose is not present, because in the absence of the lactose, the Lac repressor binds to the operator and then gets in the way of RNA polymerase binding to the promoter. If the glucose and lactose are both not present, that would force the bacterial cell to use a different carbon source. So there's something turning it on and something turning it off. So the cyclic AMP bound to CAP would turn the RNA polymerase on, but the Lac repressor is in this way. So the off signal takes priority here. So the operon is on only if glucose is absent and lactose is present. Glucose being absent will cause cyclic AMP to act as a ligand to bind to CAP, and then CAP binds to the DNA. The lactose being present, this isn't shown in this diagram, but lactose being present will cause the concentration of allolactose to go up. That binds to the Lac receptor and pulls it off of the DNA. So now CAP is able to activate RNA polymerase, and the repressor is not there to get in its way, so the operon is on, and the messenger RNA, the codes for those 3 genes, will be transcribed. So let's look at that operator a little bit more closely. It actually consists of regions called O1, O2, and O3. So what was shown in the last diagrams was actually O1. So O1 is the primary repressor binding site, and that's shown in this figure right here. And it overlaps the transcription site. So the transcription site is right here where this little blue thing is. O2 is 400 base pairs downstream, and it's actually fully within the Lacz gene. O3 is upstream of O1 at the end of that Lac gene, and remember Lac gene coded for the repressor. So you do need all 3 of those repressors for maximum repression. So let's see how this works. Well, the last figures didn't show this. They were a little cartoony, but the Lac repressor is a tetramer, and we can think of it as a dimer of dimers. So this shows this in a little bit more detail. So if we have a dimer here that binds to O1, then the other dimer of the Lac repressor can bind to O2. And again, it's not shown here, but in order for the Lac repressor to bind, it has no ligand bound. So there's no allolactose bound. So when the O1 and the O2 form, there's a little DNA loop that forms. So here, actually that's a big DNA loop. You get a 401 base pair loop because The other way that the Lac repressor can bind is that the dimer can bind to O1 and the other dimer can bind to O3, and that forms a smaller loop of 93 base pairs. So these actually can kind of switch back and forth, but notice the Lac repressor, there's always a dimer bound to O1. So sometimes the other dimer is bound to O2 and sometimes it's bound to O3. So we have some pictures of what this looks like. So this is the Lac Repressor Tetramer showed in this sort of this hot pink fuchsia color right here. And it is a tetramer, although they didn't color the subunits different colors, so we can't really see that. But this is a dimer right here, and this is a dimer right here. And this orange part right here, that's the DNA. So this shows the O1 and the O3. So this is O1 and O3. So you form this big loop right here, and notice the other things that are within the loop. The -10 and the -35 sequences are shown in green, and that's in that loop. And in blue, that's cap. So cap is binding to its site right here. So if RNA polymerase is expected to find the promoter at -10 and -35, notice that when the Lac repressor is bound and cap is bound, it would be really difficult for RNA polymerase to be able to bind to the promoter. Here is another segment right here that shows, and we can see this in a little bit more detail because here the artist did color the subunits different colors. So we have O1 here, I believe this is O2, and it shows the binding of the dimers. And you can see that this monomer right here, you can see them binding really tightly and really nicely to the major grooves, like we talked about in the last unit. And the same thing over here, we have the orange binding to a major groove, and another portion, another domain binding to the major groove over here. So to summarize, this is from a different book and it shows a little bit more of the detail. So this is where a cap can bind. This is the promoter that has -10 and -35, although it doesn't show that there. Here is the operator. Now this is O1. They're not going to show what the loops for O2 and O3 here. So that's O1. And then here is the Lacz gene right here. So this is, this orange thing is RNA polymerase, and remember it has sigma 70, and sigma 70's job is to find the promoter, the -35 and the -10 Tata box right here. But when the Lac repressor is bound, the RNA polymerase isn't able to bind. And here we have glucose, so cap's not bound, so the cell will use glucose preferentially. So what happens if you have both lactose and glucose? What happens here is the RNA polymerase again is looking for that promoter, but it's not being stimulated by cap because cap still isn't bound, because when glucose concentration is high, the concentration of cyclic AMP is low. And remember, cyclic AMP is the ligand for cap. So the RNA polymerase can bind, and the lactose repressor is not bound because now these little pink squares right here, that's the molecules of allolactose. So allolactose is bound because remember, if we have lactose, then we have some allolactose being formed. And we can explain this by saying that we have a low amount of transcription. So we have a little bit of beta-galactosidase being made, but not the maximum amount possible because we don't have cap here to stimulate the RNA polymerase. So that's what happens here. So if lactose is present and glucose is absent, now the concentration of cyclic AMP goes up, and cyclic AMP now then binds to cap, and that causes a conformational change that allows cap to bind to the DNA, and it binds in a way that encourages the RNA polymerase to find their promoter and to initiate transcription. And again, the Lac repressor will not be in our way because we have the allolactose being bound right here. That's the ligand for the Lac repressor. That causes a conformational change that causes the repressor to dissociate from the DNA. And now, with the stimulator there and the repressor gone, the RNA polymerase can carry out transcription at a high rate. Okay, so let's talk about a different system. So this is an on-off switch. This is called the Trp operon. So the Trp operon, this is the operon that the cell will turn on if it needs to synthesize tryptophan. So if you have tryptophan provided in the growth medium, there's no reason for E. coli to make its own tryptophan if you've already provided it. So here, there's the promoter, minus 35 and minus 10, and then there's the operator. So here the operator is actually in between. The genes that we have here, there are 5 genes that need to be made to synthesize the genes that are required for tryptophan biosynthesis. So here they are. So this is a picture from our book. So this shows the promoter and the operator. So I'll show you this in a little more detail in a second. We can make an entire transcript of messenger RNA right here. We can make just the leader, or we can make the whole thing. So I'll explain that in a minute. But what this shows is this diagrams the tryptophan biosynthetic pathway, starting from corazimate and glutamine and going all the way to tryptophan. So there are 3 steps right here. So for these 2 steps, sorry, the same enzyme catalyzes both of these steps. So this is an enzyme that is a dimer, so it has 2 components, component 1 and component 2. So that's made by the TrpE and the TrpD genes. So that can catalyze both of these steps. And then when you get to this step where you have the N5' phosphoribosyl anthranilate, these 2 steps are catalyzed by this synthase enzyme that's coded for by TrpC gene. And then this last step is a tryptophan synthase gene. So it's a tetramer, 2 alphas, 2 betas. So the TrpB gene makes the beta subunit and the TrpA makes the alpha subunit. So you need all 5 of these genes to assemble this tryptophan synthesizing factory. And again, you're going to do that when Trp is absent. So if Trp is absent, we're going to make this entire messenger RNA transcript. But if Trp is present, we're not going to bother. We'll make this leader messenger RNA, and then we're going to stop. So I'll show you how that works in a second. Within the promoter is the operator, and the operator has a sequence that's recognized by the Trp receptor. So I showed you this a long time ago when we were looking at proteins that bind to the DNA. So this is a helix turn helix. So we have the helices here shown as these cylinders. We have helix turn helix, and in red, that's the recognition helix. So that's the part that's going to bind to the DNA. So the Trp repressor binds to the DNA as a dimer. We've seen that before. So the recognition helix, we're going to have one portion bind here, and this major groove, and this portion will bind in that major groove. So when the Trp repressor is bound to the operator, it blocks access to the promoter by the RNA polymerase. But the only way that the Trp repressor can bind is that it needs, it's not shown here, but it needs 2 Trp ligands in order to have the proper conformation so the repressor can bind. And the Trp ligands are a signal to the repressor that Trp is plentiful, and we ought not to be expressing those 5 genes. So here's how it works. So there's the operator. It's in the middle of this promoter sequence. So here's the minus 35 and there's the minus 10 sequence. So if tryptophan concentration is low, we need to make those 5 enzymes. So here's the RNA polymerase, and it recognizes the minus 35 and the minus 10 sequence. The operon is on, and it synthesizes this long messenger RNA transcript that can be used during translation to make those 5 enzymes. In this case, the repressor is inactive. It has a conformation that doesn't allow it to bind to the DNA. But if the tryptophan concentration is high, then tryptophan acts as a ligand and binds to the receptor, and then the receptor binds to the operator, and because the operator is right in the middle of these 2 minus 35 and minus 10 sequences, the RNA polymerase can't bind, and the operon is off. So I wanted to show you again a little bit more of what this picture actually looks like. So here is showing the helix turn helix domains. In here you can actually see the helices. So you can see the helix turn helix here, and the two monomers of the dimer are shown. One of the monomers is blue and the other one is reddish pinkish. What the artist did right here is the portion, the helix turn helix motif that binds to the DNA is shown in a darker color. So the dark blue right here is shown binding to the major groove of the DNA. And over here, I guess dark pink is red. So here's the helix turn helix motif right here binding to the major groove. So we have our nice dimer, and there in a space filling model is our tryptophan. So when the tryptophan is bound, we need one tryptophan to bind to each monomer. We have the Trp repressor and the conformation that it needs to bind to the operator on the DNA. So this shows this maybe in a little bit more of a, maybe a bigger picture kind of thing, where this shows the Trp repressor. You can see that these are kind of maybe at angles to each other, and it's not able, the helix turn helix motifs that are shown in barrels aren't able to bind to the major groove of the DNA. But if Trp bounds, so this is a Trp, the ligand, and it's shown in red. So I've got a Trp ligand here and a Trp ligand here, and now there's a conformational change that occurs when that ligand binds, and now the helix turn helix motif can very easily bind to the major grooves. So this is negative control. So the active DNA binding form of the protein with the ligands bound serves to turn the genes off. So that's the definition of negative control. So the gene regulatory proteins that function in this way are called transcriptional repressors or gene repressor proteins. So in this system, Trp acts as a co-repressor. So its presence prevents superfluous Trp synthesis. You don't need to be making tryptophan if you already have enough. And having these ligands that have the opportunity to bind to the Trp repressor is a signal that the Trp concentrations in the cell are plentiful. So let's go back to that leader sequence that I showed you before. So this shows the entire Trp operon, and what I've done here is I got this from a different textbook, but I took this portion right here and I blew it up. So this is a 140 nucleotide base pair sequence of the Trp operon. So this is called the leader. And so our translation start codon is here, and then we have these 4 domains followed by a whole string of U's. So let's see how this works. The way that this works is very clever because in a bacterial cell, you don't have the barrier of nucleus. So you can transcribe your RNA and you can start translating it before transcription is done. And that's what happens here. So, so here is the messenger RNA right here. And here is our ribosome, our small and our large subunit. So here's that leader sequence. And the way that this works is right in here in this box that's labeled 1, that region 1, that has 2 tryptophan codons. So if Trp concentration is high, you'll have a tRNA with a Trp charged onto it, and you'll have no trouble going through and translating. So here's you're making a little leader peptide right here. You'll have no trouble doing this because you'll have plenty of Trp to be able to continue to do this. So, but as the ribosome is sort of chugging along doing this, this gives the opportunity for regions 3 and 4 to form a stem loop structure. And remember, we had those long string of U's right here. So this is a termination signal right here. So if you go past 1 and now you're in part 2, and then now 3 and 4 have the opportunity to form this termination signal, then transcription will terminate and translation will terminate too. So you'll stop transcription and you'll just degrade this little bit of the leader peptide. But what happens if you don't have enough trip? So if you don't have enough trip, remember region 1 has those 2 trip codons. So here you are, and you started to translate the messenger RNA, and you get to that first region, and you've got 2 trip codons, and you don't have any tRNAs charged with tryptophan in order to be able to continue the translation. So the ribosome is going to stall. So you'll start to make the leader peptide, but you're going to stall right here. So what happens is now regions 2 and 3 form a stem loop structure, but that is not a termination signal. So what happens is RNA polymerase will continue to transcribe, and you'll transcribe the rest of the trip operon. So, so this is the structures of what these things look like. So, so this is the when 3 and 4 have the opportunity to get together. So this is region 3 and region 4, and this is region 1 shown here in green. And notice right next to each other are 2 consecutive trip codons. So if you can translate those, great, and then that gives the 3 and 4 an opportunity to form this termination structure. But so this you'll have termination if the trip concentration is high. But if the trip concentration is low, so here our authors call this an anti-terminator. So you get to the point where you want to you want to translate those trip codons and you can't do it. So you have this sequence that forms instead. That's not a termination sequence. So what happens is you don't stop here. You complete the transcription of the entire messenger RNA transcript. So here's sort of a cartoon of what that looks like. So here in high tryptophan situations, here's my ribosome. I've gone through region 1. I can do that because I have the tryptophan to do that. But now the transcription terminator forms, and then the RNA polymerase dissociates from the messenger RNA, and transcription is terminated. But if tryptophan is low, the ribosome stalls right here at those 2 consecutive trip codons. The anti-terminator forms, and here transcription continues until you get to the end of the operon. So it's a very clever situation, I think. Okay, so let me give you one more example. So the 2 examples I just gave you involved regulatory sequences that were very close to the promoter. So that's not always the case. Here is a situation where we have something called Ntrc, and that's a regulatory sequence, but it's way, way upstream of the promoter. And in fact, essential to this is being able to form some kind of a looped intermediate. So this is involved in turning a gene on for glutamine synthetase. So this would happen if the cell was low on glutamine. So you could use ammonia and glutamate to make glutamine as long as you have glutamine synthetase. So if glin concentration is low, you're going to need to turn a gene on to synthesize glutamine synthetase. So the Ntrc protein is the nitrogen regulatory protein C, and it stimulates transcription of glin A. So glin A is the name of the gene that encodes the glin synthetase. So you might have noticed in the last slide that the sigma was referred to as sigma 54. So I show you this, I think I've shown you this before, I'm not sure, but we often talk about sigma 70 as if that were the only sigma, but it's not. We do talk about sigma 70 because most genes and exponentially regulating cells use sigma 70, the housekeeping genes do, and we're very familiar with the -35 sequence, the TTGACA, and the -10 region, which encodes the TATA box. But there's this whole suite of sigma subunits that the RNA polymerase can use. It doesn't always use sigma 70. So how about now we use an example? Let's talk about this sigma 54. Sometimes it's called sigma N because it regulates the genes involved in nitrogen metabolism. So we have a -35. Oh, sorry, here it's a -24 region and a -12 region. So this is analogous to the -35 and the -10, but it's located in a slightly different place. So let's see how this works. So again, just to remind you, glutamine synthetase is the enzyme that we need if we need glin. So glin is in short supply. We can make it from glue and NH3. So NTRC is a dimer, and it needs to be phosphorylated, and it's phosphorylated when glin levels are low, and I'll show you that in a second. So we have dimers, but we have a dimer of dimers. So here the P means that this thing is phosphorylated. And again, it is way upstream. But once this thing is phosphorylated and it's able to bind um, to DNA, then what happens is it forms this kind of like loop signal right here, where here's the RNA polymerase that has a sigma 54 associated with it. So the NTRC is able to associate with this RNA polymerase with its sigma 54 and able to encourage it to transcribe the glin A gene. So this is how it works. So when glutamine concentration is high, you don't need to make that enzyme because you have plenty of glutamine. So if glutamine concentration is high, that acts as a ligand and it binds to NTRB. So that's shown right here. So this is glin and it's bound to this sensor domain on NTRB. And there's a kinase right here, and when the glin is bound to the sensor domain covers up the active site so the kinase is inactive. But when glin levels fall, then the glin dissociates from the NTRB. So you have this conformational change that occurs, and now it looks like this. And now there's a histidine right there that is now exposed, and it can be phosphorylated. So ATP is the phosphate donor. And then the next thing that happens is the phosphate is transferred from NTRB to NTRC. So here's NTRC, and it has a regulatory domain and a DNA binding domain. And when the regulatory domain is not phosphorylated, the DNA binding domain is covered up. But when this His on NTRB gets phosphorylated, it transfers the phosphate to an aspartate on the regulatory domain. And now imagine this kind of swinging out right here. And now the DNA binding domain is exposed, and now DNA binding domain can bind to its enhancer sequence, and then it can interact with RNA polymerase with its sigma 54 and encourage RNA polymerase to transcribe the glin A gene, which will code for glutamine synthetase. Mother Nature is so clever. Here are my references. So I hope you enjoyed hearing all about prokaryotic gene regulations, and see you next time.